Hi guys, so in this video I'm going to talk about the genetic code and how to use the codon circle to determine what amino acid a sequence of DNA or RNA is providing the instructions for. So essentially a codon are three non-overlapping nucleotides. We could be referring to either the original DNA sequence or the temporary copy stored in mRNA. And each codon is going to specify a single amino acid. So when we're reading this codon circle up here, we start off in the middle and then we work our way out. So for example, if the sequence is GAG, that codon is going to bring the amino acid glutamic acid to our sequence. There are a few special codons that we have to make sure that we remember. The first is called the start codon, and that is always going to be AUG. So the start codon here will always bring the amino acid methionine to the beginning of the sequence. Essentially, that's how the ribosome knows to start the process of translation. There are also three stop codons. So if we look over here at the upper right corner of the codon circle, the sequences UAA, UAG, or UGA are all going to code for what it says here is stop. So obviously that's not an amino acid. Essentially what that is doing is telling the ribosome to stop synthesizing the protein sequence. So let's do a little bit of practice here. On these notes, you have a sequence of DNA, and I can tell that this is DNA because there are T's and not U's. And we're going to write out the mRNA sequence first, starting with our template strand. So in this sequence, you're told that the bottom strand is the template, and it also tells you that the first four nucleotides there, A, T, A, T, are part of the promoter sequence. So that doesn't get transcribed to mRNA. We're going to start directly after the promoter. Now, if the bottom strand is my template, whatever I'm building off of that is going to be anti-parallel, meaning that the bottom strand is 3 to 5. We can see here it starts 3 and it goes to 5. So the complementary sequence of mRNA that's going to bind to that will start off at the 5' end. Like I said, we're going to start right after the promoter region. So if there's a T in the DNA sequence, that's going to bring an A into the mRNA sequence. An A in DNA is going to bring a U into the mRNA sequence. Remember, there are no T's in mRNA. And then the C right here is going to bring a G. And then I can keep going using those same methods. So then we have G, G, U, A, U, C, G, A, U, U, A, and that will bring us to the 3' end of that mRNA strand. The next thing that we're going to do here is add in the protein sequence. And we're going to use the codon circle to determine the protein sequence from this mRNA sequence. So remember, we read 3 nucleotides at a time. So our first 3 nucleotides are A, U, G. So using the codon circle, we start with A in the middle and work our way out. U, G, that's methionine. Remember, that's always going to be our start codon. So if we're given an mRNA sequence that doesn't begin with AUG, we have to scan the sequence to find that in order to begin the translation process. Okay, the next 3 nucleotides here are G, G, U. So going back to my codon circle, G, G, U is going to give me glycine. Then we have AUC. So AUC gives me isoleucine. Then GAU. Up here, GAU gives me aspartic acid, ASP. And then UAA over here gives me a stop codon. So UAA means stop. So I'm not actually going to write anything in at the end of the protein sequence. I just know that that's the end of that sequence. So that's essentially how we are transcribing and translating the genetic code that is initially found in DNA to mRNA and then finally to the protein sequence. I hope this video was helpful.